students. My name is Dr. Salahuddin, working as Associate Professor in Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Today, I will discuss about the solution phase synthesis, that is the another method involved in the combinatorial chemistry. In the previous lecture, I have already discussed the first part of this combinatorial chemistry, that is the solid phase synthesis. In this lecture, I will discuss about the various aspects about the solution phase synthesis. In the solution phase, in the combinatorial chemistry, or the we can say it is a CombiChem, is an innovative method for the synthesizing a different substance quickly at the same time, and here combinatorial chemistry contrast with the time consuming and labor incentive method of the traditional chemistry, where the compound synthesized individually at one time. So, combinatorial chemistry used to synthesize a large number of the chemical compounds by combining the sets of the building blocks. So, here for the building blocks, we are synthesizing the new compounds, a new composition of the compounds which are slightly different from the previous one. Whatever the compounds that we are synthesizing, the second compound must be different from the previous one. So, we can synthesize the different types of compounds with the help of this combinatorial chemistry. Here we will discuss about the solution phase synthesis. So, when the combinatorial chemistry first emerged, the initial focus was the solid phase approach due to the many advantage. So, and here this solution phase chemistry was not regarded as being suitable for the combinatorial chemistry because of the often tedious isolation and purification. So, here in this solution phase synthesis, the isolation and the purification, in every chemical reaction, isolation and the purification, these two steps is very important steps, because when we are synthesizing the compounds, there is a major chance to develop the impurity, or sometimes the phases will separate out. So, the two phase comes. So, the isolation isolation will be very tough. Sometimes the isomers of the compounds develops. So, we have to separate out these two different types of compounds. So, here the main problem of the solution phase combinatorial synthesis is to obtain the pure products. The significance of the purity varies with the purpose for which a combinatorial library is produced. Search for a new ligand versus lead optimization. So, here this is also involves the searching of the new ligands and the searching of the new lead compounds and the lead optimization. Next thing is what? Reaction proceed in the solution. That is why the name indicate solution phase synthesis. Faster validation, time related to the solid phase synthesis, as the time required for here that is the solution phase synthesis, it is less time required from the solid phase synthesis in the solution phase. So, here the standard analytical protocol can be used to characterize the products between each reaction step. So, as the conventional synthesis and the combinatorial synthesis, in both the cases analytical protocol can be used to characterize the product. Whatever we are synthesizing the compounds, we have to characterize at each step, either the compound is properly synthesized or not. So, characterization is very helpful to remove the impurities, and it will again it will helpful to synthesize in the next step. Easy characterization of the intermediates as well as the end product. Here there is no limitation in the attachment point. Difficult to drive the reaction towards the product. Extensive purification is needed. Now, here in the solution phase synthesis, there are the various protocol that we have to follow, that reaction proceed in the solution, reaction proceed in the solution. So, here the solution is required as the name indicate. It can be produced to produce libraries that consist of the single compounds or a mixture of the compound. This solution phase synthesis that we can apply for the single compounds and the mixture of the compounds in both the cases. Now, here the easy characterization in between the steps also. In between the steps as well as the final products. Next is a standard analytical protocol to characterize the product. Here, what is the basic difference between the solution phase chemistry and the solid phase chemistry, or the possible strategy in the combinatorial synthesis? Here are some different types of building blocks that we are using. Different small building blocks that we are using, and these are the building blocks also present here. There is no different in the building blocks, but only the difference here that we are using the solution, and in the solid phase that we are using this solid phase solid support in the form of resin. The different types of resin for the different type of product that we can use in the solid phase chemistry. Here I am discussing about the solution phase synthesis. So, here are the some parameters we can understand by taking some parameters, that what the parameters that we are taking in the solid phase and what parameters that we are using in the solution phase. First thing is reagent. As I have already told in the previous lecture that we can use the excess reagent, and this excess reagent that we can remove after the completion of the reaction. Here in the solution phase we cannot use the excess reagent, we can use the optimum solution, that unless the purification done. The purification in the solid phase is easy, and here the purification is difficult because we are using here the solution. Automation, here the automation is easy and the automation of is difficult. So, here the combinatorial chemistry based on automation process. Reaction suitable for the few substance, and here so suitable for any organic reaction. So, here the application of this solution phase that we can apply in any organic reaction. If we scale up the solid phase, that is expensive and it is easy and inexpensive. Here there is no solid support, no linker, generally we are using the only the solution. Next is dependence of the reaction, solid support linker. Here the reaction is dependent on the solid support that we are using. Reaction is dependent upon the linkers, but in the solution phase synthesis the reaction is dependent over time. So, it is a time dependent reaction, that is the solution phase reaction. When we are planning a combinatorial synthesis, whatever we are planning to do something, we have to prepare something. So, here before doing this combinatorial synthesis, our aim is not to synthesize any type of molecule. Our aim is to produce focused or targeted libraries. So, the target molecule should obey Lipinski rule of 5. Lipinski rule. Lipinski rule for oral activity. So, this Lipinski rule, molecular weight less than 5, molecular weight less than 500 molecular weight, log p value that is less than 5, hydrogen bonding donor and hydrogen bonding acceptor. These 4 properties is very important. If any ligands having all these properties, molecular weight less than 500, calculated log p is less than 5, hydrogen bonding donor is 5, hydrogen bonding acceptor atom is 10, then we are able to say that the compound must be act as a lead compound. So, here the when we are planning a combinatorial chemistry, our targeted ligand follow this rule. Now, we have to choose the scaffold. So, the scaffold is based on either tadpole scaffold or privileged scaffold. Tadpole scaffold is restricted to a specific region round the molecule, the specific molecule that is required, and there is less chance of favorable interaction with the binding site. In the privileged scaffold, which are common in medicinal chemistry and which are associated with the diverse range of activity. First, we have to choose the scaffold. Scaffold means the basic structure, the small structure like benzodiazepine, hydantoin, benzene sulfonamide, etc. Now, here the various other scaffold, that is the benzodiazepine, here hydantoin, that you can use this hydantoin as an anticonvulsant, beta lactam, antibiotics, some pyridine derivatives is anti-cancer, dipeptides also, and these are the good scaffold that we can use this scaffold. Spider like structure, that is the low molecular weight, variety of the synthetic route available. We cannot choose that type of scaffold that the synthetic route is not available. So, we have to choose only those route which have the ability to synthesize. Now, the poor example, that is the glucose, spider like and a small molecular weight, but multiple OH, different type of OH groups are present in the glucose, and the various different types of the that is the R1 and R5, that is independently. Next is scaffold, that is the steroid, that is molecular weight is relatively high as we know, and restrict the number of functional group to keep molecular weight less than 500. So, if we are choosing the steroid moiety, their molecular weight is it should be molecular weight less than 500, and it is very difficult to organize the molecular weight is less than 500. So, that we have to restrict our functional group to reduce the functional group. So, here the relatively few points where the substituents that we can easily add. Next is indole. Indole is also comes in the under the poor scaffolds. Here it is a because it is a tadpole like scaffold, restricted region of variability. Now, in the isolation. So, first upon the when we are doing this solution phase, we are doing the we can do the isolation with the help of recursive deconvolution, micro manipulation and the sequential release. After the isolation, we can do the structural determination of the active compounds. Structural determination with the help of tagging, with the help of encoding sheets and photolithography. Now, here this is the very interesting slide for the solution phase synthesis. So, if we are synthesizing the amide derivative using this RCOCl and amine. So, here the first thing that we can take this acid chloride based, and the second thing is amine based. So, here at here A1 means first acid chloride like CH3COCl, and B1 to B10 means 10 different type of amine. So, 10 different type of amine are NH2. So, you have to dissociate, you have to substitute with different type of this amines. So, this acid chloride set we can produce a mixture one containing all possible A1 to B compound, means A1 B1, A1 B2, A1 B3 like this, A1 B1, A1 B2, A1 B3. So, that it will produce a different type of compound. Next thing, like here it will produce the 10 different derivatives. So, like this here we can produce the number of various number of derivatives by using either first thing is acid chloride set and the amine set. Next is what is the recursive deconvolution, method of identifying the active component in the mixture, quicker than method of identifying the active component in the mixture. So, if there is a different type of mixture is obtained in a vessel, so that we have to identify the active component by using this recursive deconvolution. It is quicker than separately synthesizing all possible components, that we have to need to retain the sample before each mixed and split phase. For example, all 27 tripeptides by mix and split strategy by glycine, alanine and valine. So, here we are using the three different type of amine that we are using, that is glycine, here the glycine, alanine, alanine. First you mix it and split it, mixed and split. So, what happened? We have mixture 1, mixture 2, mixture 3. So, in the mixture 1 have glycine, valine, alanine, mixture 2 also have glycine, valine, alanine, mixture 3 also have glycine, valine, alanine. Now, add glycine in this first mixture. So, it will produce the next reaction, next mixture, mixture 4, 5, 6. So, again the entry of this glycine, entry of this alanine in this mixture, entry of this valine at here. So, all possible dipeptides in 3 vessel retain a sample from each vessel. Now, in the mixed and split method, first then again you have to mixed and split method, then add mixed and split, then add glycine, alanine and valine. So, you will get a different type of tripeptide. First you will get dipeptide, then you will get a tripeptide. Again this mixture is inactive mixture, we will get mixture inactive and this mixture is active. So, that the 9 possible tripeptides in active mixture and all end in valine. Add valine to the 3 retained dipeptides. So, we can add all the all in this inactive mixture, that is valine, it produce the another active compounds here. So, we have the different active component narrowed down to one of the 3 possible tripeptides. Synthesize each tripeptides and test. Now, here is the detection, purification and analysis. So, detection at each and every steps. So, that we have to procure the compounds each and every step for the detection of the compounds, and we can analyze, we can quantities of the analyzed compound are very small, non-destructive. The process of inspecting, testing and evaluating material components or assemblies for the discontinuous. Whenever we are synthesizing, if it is discontinuous, then this comes under the dense non-destructive. Rapid or parallel analysis, use analytical techniques like high performance liquid chromatography, mass spectrometry, chromatography, most importantly IR, computerized methods. We can use NMR, 1H NMR, we can use 13C NMR, we can use this HPLC NMR, mass spectrometry for the characterization of the compounds. And next is supercritical fluid chromatography. So, here the chromatography and spectroscopy are used for the detection or the purification of the different type of compounds that previously produced from the solution phase techniques. And this is the some application of the combinatorial chemistry. The most important application of this combinatorial chemistry is our requirement, that is the computational chemistry, and it is involved in this QSAR, QSPR both, and the rest is the various different type of application of this combinatorial chemistry, including the solid phase synthesis and the liquid phase synthesis both. Thank you so much.